Okay, so first I would like to thank the organizers for giving me the opportunity to talk my research here. Again, the second is that I want to thank you all for still staying here. So today I'm going to talk about complex genetic interactions and the COVID-19 severity. So let's first start with single variant based GWAS studies. So here's a summary of summary part I summarized from 18 COVID-19 studies. Oh, I'm sorry. So just
People all over the world.
okay, sorry. Okay, sorry. I'm just so used to face the screen. So anyway, so basically this is a summary table to show the existing studies, the genetic variant discovered by current studies. There are about 85 unique SNPs, and they can be mapped to about 60 genes. From here, you can see there are some multiple genes that actually can be frequently discovered by multiple different studies, which is very inspiring. So now the question is that, are we able to use these variants discovered to build a predictive model? So the answer probably is a no. So we know that traditional GWAS study is based on single variants, so which means they assume all the variants they are independent from each other. However, COVID-19 is a complex disease. So there's an instance that the effect of one mutation might be actually depends on the mutations from other variants. So for example, ABC and XYZ are two pathways. They share some similar functions, and this function can be associated with the disease risk. And then we found that because they share some similar functions, when the mutation is found in one pathway, the other pathway can compensate the loss of function. So basically you are not going to cause loss of function. Only when the mutations happen on both pathways, you will see a loss of function. Then we call there's a genetic interaction between gene A and the gene Y, something like that. However, in GWAS study, genetic interaction is mostly understudied because of lack of statistical power, because there are too many variants, right? So my work for the past 10 years, I'm trying to study genetic interaction from GWAS because there are so many data there, and we just do a single association analysis. I think is a big waste of data. So we developed a bridge method, which is a pathway-based analysis tool for detecting genetic interactions from GWAS. So I show here is that we have two publications. We actually apply this method to breast cancer, which is published in PLOS Genetics in 2017, and also we have apply this method to additional 6 diseases in our 2019 Nature Communication publications. So more specifically, we are interested in two types of pathway interactions. One is called between pathway model, another is called within pathway model. When we say pathway ABC and the pathway XYZ has an interaction, which means the SNP level interactions within this, between these two pathways, they're going to be enriched for SNP level interactions. For the within pathway models, which means if a pathway has within pathway interaction, then the interaction within this pathway are enriched for SNP level interactions. So, I only have 15 minutes, so I'm just going to have a very brief introduction of this method. So the input of bridge is, GWAS data set and also a set of pathways. And we do data processing, and then we compute pairwise SNP interactions. Then we construct a SNP interaction network based on different disease model. So, and then from there, we can, depending on the association, the interaction is associated with increased or decreased disease risk. We split the network to a protective or a risk interaction network. And then we take the pathways, and then we evaluate BPM and WPM. Basically, we try to evaluate if the the interactions within the structure are enriched for SNP level interactions. Then we repeat this process. We shuffle the sample labels for 100 times. So then we evaluate the false discovery rate for the BPM, WPMs. So the output of our method is going to be significant pathway level interactions and also driver SNP or genes for that, for that pathway. Now we are going to talk about COVID-19 now. So we use UK Biobank as our COVID-19 data resource. So the UK Biobank collected data from England, Scotland, and Wales. And basically, the last column of this table, you can see the number of COVID positive inpatient. Basically, this is our case group. That these are the COVID-19 severe cases. We also using controls. The controls we use is COVID-19 positive. They are not inpatient. So basically, we are just like, all the samples have COVID positive, but the one is like have very minor, basically maybe no symptoms, but the cases they need to hospital care. Then we use England data for discovery analysis and Scotland and the Wales data for validation analysis. The pathway we use are from M6DB, and after filtering for redundancy, we have about 1,400 pathways. Now we apply this, we apply a bridge to the England cohort. We find at a false discovery rate less than 0.05, we found 77 BPMs and 12 WPMs. They all associated with COVID. Actually, they also associated with increased COVID-19 severity. Then we require all this discovery need to be replicated in both Scotland and Wales cohort at a p-value less than 0.05. This actually in the end give us 33 BPMs and 8 WPMs. So I plot this BPM, WPM in terms of pathway interaction network. If you guys can see the name of these pathways, you will be able to find that a lot of this pathway is associated with immune response, inflammatory response, or viral infection pathways. So which means that our discovered pathway are very relevant to COVID-19. So more specifically, I want to give you two examples. These are the two hub pathways in our interaction network. One is this interferon gamma signaling pathway. So basically, previous research, they have found that they actually find higher interferon gamma levels in the patients who died than the survival patients. So this is a very strong support for our discovery. Another interesting story is that the androgen signaling pathway regulates the SARS-CoV-2 receptor levels and is also associated with COVID-19. So basically, I just give two small examples, but many of these pathways we discover are really relevant to COVID-19. But I want to move on because they are more interesting things to discuss here. So, so one way we can use our discovery is that we can provide these pathways as candidates for, for biological, evaluation. But I'm a computational person, so what we can do with this result? So here, I want to give an example. So basically, you already have concept about polygenetic scores here. So I want to just introduce a new concept, which is a genetic interaction-based polygenetic risk score. So for example, the left is actually a visualization of BPM, which is between the interferon gamma signaling pathway and the androgen receptor. So the left is basically the left and right, there are two pathways on each side. The edges between these two pathways are interactions. And the bars I'm showing here, basically I'm showing their single variant association. So one inside you can see majority of the variants we found in our BPM or WPM, they don't reach genome-wide significance. Okay, so now start from here. For each age in this BPM, we can find that what's the genotype it contributes to the interaction. Then we assign for each individual, we can assign, one if this individual have this genotype, 0 if he doesn't. We can do this for all the SNP pairs in this BPM structure. In the end, we summarize this. We summarize this number, just tell you if a patient, if an individual have a higher number of, SNP pairs, they are related to the interaction, they have a higher risk to getting the disease. In the end, we normalize these numbers and get a genetic interaction-based PRS scores. So from this box plot, you can see our PRS score actually kind of can differentiate case and controls from this England data. So think about this is just the 1 BPMs. We have 77 BPM and 12 WPM from our discovery. So basically you can direct think about maybe we can, if we put using all the discovered BPM, WPM together, can we build a prediction model for COVID-19 severity? So this is our goal. So to do that, first we want to define a base model for comparison. So age is the best predictor for COVID-19 severity. So we use our base model is going to be age and gender. Oh, 1 minute. Oh my gosh, sorry. Okay, so basically, I have like basically here, I want to tell you that I have a single variant based prediction model and also genetic interaction based prediction model. I can show that the single variant based prediction model can increase the prediction, improves about 5%, but our genetic interaction based prediction model can increase prediction by 20 to 35%. But this is from the original England data. So the more harder question is that is this model can be generalizable to independent cohort. So we apply this model actually to the Scotland and the Wales cohort. Which are independent. The model is trained on the England cohort, and the features are selected from the England cohort. But we are able to show that it actually can, our genetic interaction-based model perform much better than the single variant-based model. And this kind of performance in an independent cohort is very rare for GWAS data set. And then in the end, in the summary, so we discovered 33 BPMs and 8 WPMs from the UK Biobank data. And also the majority of pathways discovered are novel when compared to traditional single variant based approach. We also show that genetic interaction predict COVID-19 severity better than model-based single variants. We also suggest that the complex genetic interaction between common variants play an important role in determining COVID-19 severity. And I also would like to thank my PI, Chad Myers, and also the two students, Meret and Matthew, are the two students working with me for the bridge-related work. And I still want to say we have a virtual poster, which is about the genetic interaction-based machine learning models. So if you're interested, you can search the poster. Thank you all.
So do we have questions for Wen?
So a lot of these pathways are sharing genes.
Yes.
How do you know that SNPs that are inserted on one gene that's sharing multiple pathways not joining?
Yes, you're right. So basically, I'm just saying you're saying some of the pathways they can share same amount of genes or SNPs. I think when I designed this, when I evaluate the basically if the two pathways are interacting with each other, I exclude the common variants or common SNPs between the two pathways.